Welcome to today's BIOS Roundtable. We are thrilled to welcome two of TechBio's foremost leaders to the show today. Thank you for joining us, Daphne Koller, CEO at Insitro, and Chris Gibson, CEO at Recursion. We're thrilled to host you both and excited to embark on today's episode. Joining me to co-pilot this episode into the future along this voyage, we got Chris Godbon, my great colleague here. We'd love to kick things off, folks. Can you please give us a quick introduction on yourselves? Daphne, can you give us a good overview on yourself and Insitro, please? Thank you.
So I have a somewhat unusual career journey that started out pretty traditional. I thought I would have a career as an academic and would retire as an academic. I was early to machine learning back in the early 90s, was hired into Stanford's computer science department as the first machine learning hire and really helped build that group over there. And then became interested in machine learning in service of biology and medicine in the late 90s as the early data sets in biology were starting to emerge. Initially, it was mostly for reasons of technical interest, but then over time, I became interested in the field in its own right and ended up in my time at Stanford having a bifurcated career where half my lab did core machine learning published in traditional computer science venues, and the other half did biology and medicine published in, Nature, Science, Cell, those kinds of journals, and thought that that would be my career going forward. And then around 2010, 2011, I began to have an increasing sense of urgency to do something that had a more immediate impact on the world, which is what ultimately led to what was supposed to be a temporary, but ended up being a permanent departure from Stanford to go and found Coursera, which is even today, I think, the largest or one of the largest online education companies in the world, over 100 million learners worldwide, and really proud of the journey that I was able to have there, despite the fact that it had absolutely nothing to do with my core research agenda. It emerged from a passion project that I was doing at Stanford on technology-assisted education. But then around 2016, I felt like I'd done what I came to do. The company was on a great trajectory. By that point, I'd resigned my Stanford position and wanted to come back and do some science and also some machine learning, because by that point, it became clear that machine learning was changing the world. And what was also clear is that it was having a much greater impact in multiple sectors of the world, but less so in the life sciences. And I believe and still believe that one of the biggest obstacles to having that impact is the relative paucity of people who are fluent in both languages, who really speak machine learning and really speak biology and can bridge the chasm between those two worlds. And so that really led me to come back to the life sciences and ask, what can machine learning people do within the life sciences to truly shift the trajectory and bring benefit to patients. And I spent 18 months at Calico. It was a wonderful experience learning about drug discovery, which I'd never been really exposed to before. And then after that, in early 2018, I decided to kind of go out on my own and build what I really wanted to build, which is an engine for doing drug discovery differently. And so segueing from my personal history to what Insitro is all about, Insitro is a machine learning enabled drug discovery and development company. And what we try and do is really bring together two complementary ways of looking at human biology and really unraveling the complexity and heterogeneity of human biology and human disease. One of those is cell-based systems that we have built that really leverage a set of tools that life scientists have developed in the last decade, induced pluripotent stem cells that recapitulate certain aspects of biological cells from a human, but in an in vitro setting, but still maintain human genetics, which for us is like the kind of North Star, if you will. Multiple ways of looking at those cells, using imaging, using transcriptomics, editing those cells, using CRISPR, and understanding what disease looks like in high content phenotypes using machine learning in an in vitro system. The other side of it is that while it's wonderful to be able to kind of look at and fix phenotypes in cells, what you ultimately care about is fixing phenotypes, disease phenotypes in people. And so the other source of data that we use is from large human cohorts where we have genetics and, again, high content phenotypes, MRI, histopathology, blood biomarkers, and using machine learning to interpret those phenotypes, and then machine learning and genetics to really bridge the gap between the human and the cell system to create a translatable in vitro system of human disease, where we can do experiments in vitro that read out on what we expect clinical outcomes look like. So that's what Insitro is. We're about 4 years old now, about 180, 190 people. We've raised $640+ million from investments, plus another large amount of money from partnerships, and we're really looking to make a difference to patients.
Brilliant. Thanks, Daphne, for joining us. Welcome to BIOS Roundtable. We're thrilled to host you today. I'll turn it over to Chris now, if he can give an introduction on himself and what he's up to at Recursion. Thank you.
Absolutely. Yeah, thanks for the chance to be here. And Daphne, fantastic to be on another podcast or webcast with you. I know we do these from time to time. So Chris Gibson, co-founder and CEO of Recursion. My introduction is much simpler than Daphne's because my pedigree is much less impressive. I was working on an MD PhD at the University of Utah, trained in bioengineering and business originally, and in that work, in the first several years, we studied a disease called cerebral cavernous malformation, trying to understand how specific genes modulated vascular stability, and ultimately figured it out, or so we thought. And as we further explored what we thought was this very promising hypothesis about the pathophysiology of this disease, biology did what it does very often. It humbled us. And it turned out that we were wrong, or we think we were wrong in our understanding of the disease. And out of that failure grew Recursion. And Recursion is really a company founded back in 2013 that's seeking to leverage many of the same technologies that Daphne just mentioned. We do cellular experiments at very high scale. We generate a variety of omics data sets from those and use those data sets to try and build maps of biology, maps of cellular biology, and now increasingly into animals and other sorts of biology as we go into the clinic with 4 programs in clinical trials now as well. So we're really a company mapping biology to try and understand relationships that perhaps we've missed using the reductionist tools of the past, so that we can give rise to new hypotheses and hopefully discover, develop medicines that'll make an important difference for patients.
Wonderful. Thanks again for joining us, Chris. Welcome to BIOS Roundtable, and looking forward to hosting you both today for a fun episode into the future of AI-enabled drug discovery. With that in mind, I'll pass it off to my co-host, Chris Godbon, to guide us through our first topic here. Thank you.
Thank you, Chaz. Thank you, Daphne. Thank you, Chris, for joining us. Recursion and Insitro are among the companies that gave rise to the founder-led tech bio movement that I think we all hope is helping drive a paradigm shift in the way we think about bioinnovation today. So Chris, let's turn first to you. How has the shift towards platform companies and the rise of tech bio really contributed to this emerging change in founder dynamics across the landscape?
So yeah, thanks for the question. I want to kind of harken back to other tech bio movements, and in particular look at companies like Genentech that really created a whole new field of biology, and in many cases were founder-led. I don't think this is totally new, but certainly in the context of technology meets biology, I think we're seeing a resurgence of these platform companies over the last 7 to 10 years, and a very large number of them. And I'm sure we'll get into it a little bit later. Some of them probably higher quality than others. But it's a really exciting time, I think, where tools in the software space have been really democratized, and we see software companies that can be built relatively easily. Obviously, building a company is never easy, but the tools to build the company are broadly available. And we're starting to see a lot of those tools be available on the biology and chemistry side, not just through CROs, but through cloud laboratories and even just the automation equipment that you can put together in a laboratory today that would have been really reserved only for very large biopharma companies maybe a decade ago. Tools like CRISPR, et cetera. And so as all these tools converge, it's creating an opportunity for folks to get started perhaps with a little bit less experience and or a little bit less capital than they would have needed a decade ago. And that's creating a lot of opportunity, and it's super exciting. In fact, there's several companies that have grown out of former employees of Recursion. And it's my favorite way to lose a good employee. I never like to lose a good employee, but my favorite way to do it is to see them go take on a mission that's really important to them. And there's companies like Inveda, Known Medicine, Noetic that are all kind of spun out in some way of the culture of Recursion. And I think it's really exciting. It's representing, to me, an inevitable revolution of new companies that are questioning a lot of the assumptions of the past. And certainly there will be mistakes. I know there's an awful lot of learnings that we've had at Recursion over the last 9 years. But there's going to be a lot of success too, and we're going to push the field forward. Insitro, Recursion, and many other companies.
That's a great message, and I really take your point around both learning and I think the culture that you build and how that ideally perpetuates forward with future newcos. So Daphne, would love your perspective on what Chris has shared. What should we think about this expanding field of life science, tech bio players, and occasionally geographies as well?
So first, I entirely resonate with what Chris so eloquently described. I think he captured it really well. There is this incredible democratization of tools across both the life sciences and compute. And Chris spoke to the life sciences, so maybe I'll speak a little bit to the compute, which is when I grew up as a machine learning scientist in the early 90s, it took you 6 to 9 months to write machine learning code that actually worked. And then if it was anything but a simple calculation, it would take you 6 months to run it in your local cluster, in your university environment, and assuming that there wasn't an electrical outage or some memory leak, then the thing would actually run and you would never run it ever again. And now we're in a world where with the tools, the software tools that are available, you can write really sophisticated machine learning code in a matter of a couple weeks and then run it in the cloud in a matter of 6 to 24 hours, and you get results back. And what that enables is not only democratization, but it also enables a rapid iteration cycle that allows you to interrogate your results, see what worked and what didn't, and then try it again. And I think that taking that back to the democratization of biological tools that Chris alluded to, this is now an opportunity to bring back some of that mindset into biology, because if a biology experiment that used to take 3 years can now take you, it's not going to be 3 hours because cells do need to grow and such, but if you could do that experiment in 3 weeks rather than 3 years, because you have all of the reagents and all of the automation and everything already in place, not only can you do a lot more stuff and get stuff done faster, you could also, really importantly, iterate on what you're doing, see what worked and what didn't, and then say, I'm going to do that experiment better, and maybe get a more informed perspective, which does what is really important, which is de-risk the biology, because ultimately it's really failure that we're looking to improve, the failure rate. So I think this really creates an incredible opportunity for life science entrepreneurs, in the same way that the democratization of computational tools has created a similar ecosystem for tech entrepreneurs. And I think that's one of the big shifts. Now, while I think there's obviously room for many different kinds of companies with different types of leadership structure, including the more traditional venture creation, or adopting a technology from academia that has scientific founders that stay in their academic roles, and you bring in whatever a hired CEO, I think there's something that is actually quite magical about a founder-led company. And I've seen many more of those in the tech space, of course, where it's much more traditional to have founder-led companies. I think that there's a certain level of passion and commitment, and this is your baby, that is just hard to replicate in any other context. And I think it brings a level of energy and just like mission-mindedness to the company, which is, I think, pretty unique. And especially in those companies that I think really require a different type of build, that are less maybe cookie cutter, less templated. A lot of that vision, not only on the strategy, but also on the culture, really comes from the founder. And I can tell you that at Insitro, and I think I've heard Chris make similar comments about Recursion, it's really about bringing together groups of individuals that normally do not communicate, that have this a huge air gap in how they think about the world, the jargon, even just like the perspective they bring to a problem. And what we have done at Insitro is really create a culture of these groups come together and they engage with each other. And this is one of our values, that you engage with each other openly, constructively, and with respect. And we see that as a core tenet of how we operate. And I can tell you that this is something that was really important to me because of how I grew up and the kind of, things that I saw working and not working in the journey that brought me to Insitro. And I think it's partly because of that this is such a core foundation of how we operate in the company.
For those who can't see it, Chaz is clapping on the back end as he should be. That's a phenomenal description, Daphne, and I know Chris and I are clapping with him, describing company culture and how building those companies is so important. Chris, do you want to add something?
Yeah, happy to. I think culture, as Daphne mentioned, is absolutely critical. And she talked about the importance of bringing folks with diverse perspectives and backgrounds together, be that folks with different social perspectives or folks with different trainings, and creating sort of a fluency and a language that we can all speak together is, frankly, I think one of the biggest moats of companies like ours, at least from my perspective. It's very difficult to recreate that in the context of a company that maybe has not had that from the beginning. And I'd just say that I think a lot of folks, I underappreciated the cultural elements in the beginning. I mean, the thing that's been hardest is not the science. What I love about science is that there's truth, but learning how to grow and lead teams, to manage people, to hire well, these are all things that I think many founders, at least first-time founders, perhaps underappreciate. And for me, the first 2 or 3 years was as much about learning those things, which is a constant development even today, as it was figuring out all the science in the early days. And so I think, my advice to founders as they move into this space is to listen to what Daphne said, to listen to what I'm saying, and pay really close attention to the culture. Because if you don't shepherd that very well, it will get away from you. And I think Daphne's right. This is one of the unique differentiators of founder-led companies. They tend to be pretty particular on these points, pretty fierce on these points. And that matters. That matters a lot. And I think it can steer companies in the right direction or the wrong one, but it certainly steers them in a clear direction.
Let's maybe even take that one step further, and this is open for both of you. Do you have any advice for founders, first time or otherwise, who are seeking to build that sort of interdisciplinary culture?
Chris, would you like to start, or?
I'll give a very brief one, Daphne, and then turn it over to you. The advice that I would give is be really open and really honest about where your blind spots are, and find people who complement them early and put them around you. So I've got lots of blind spots, lots of weaknesses, and I try to improve on those things over time. But I think one of the things that's been done well at Recursion that's made us successful is that I've tried to place people with really, really different complementary strengths around the executive team, folks like Tina Larson, who grew up at Genentech, went to Recursion, is now our president, chief operating officer. She and I see the world in extremely different ways from a tactical perspective, but I think we share a similar long-term direction. And so when you have somebody like that, it's extraordinarily helpful, although sometimes very frustrating. And I mean that in a really good way. And my advice would be to push really hard to put yourself in that uncomfortable circumstance, because if you don't do it, I think you'll find that you'll run into a lot of walls that you could have avoided.
Building on what Chris said, which I completely agree with, I'll take it in 2 different directions. One is a mistake that I made in the early days at Coursera, which is not realizing just how important it is to have on your side people who have been there, done that, because it's very easy to kind of, with the hubris of a young founder, to say, hey, I got it covered, it's all good, or even just not to realize just how much you don't know. And I think bringing in people with more more experience, especially in areas where you don't have that type of skill set, is absolutely critical and is oftentimes the difference between success and failure. The other thing I would say is, and this speaks more directly to the point of the part of the original question, which is, if you're trying to build companies that really bridge that very large gap of cultures between very diverse groups of people, how do you do that? And there's two tactics that we've put in place to help address that. One is that cultural norm that I spoke about earlier, which is how we engage with other people, the willingness to be open and vulnerable in asking stupid questions. And similarly, the willingness when someone comes with a naive suggestion that might look on the surface to be complete nonsense, like, why don't we do X? And it's like, instead of saying, well, because that's not how it's done, it's like, well, why don't we do X? And think about that, because some part of the time that might be like, why don't we do X? That's a good idea, and maybe take it from there. And I find that when you bring people who have those very distinct perspectives and have that mindset versus the protective mindset of, I know how it's done, you end up actually not only just coming up with better answers, you end up asking better questions. And so that to me is really a one critical piece to building that kind of cross-cultural bridge. And the other is, to the extent that you can find them, and they're kind of rare, not quite unicorn rare, but still pretty rare, people who are in the middle, who are bilingual, who speak both languages, so that they can play the role of integrator and translator across those different disciplines. Over time, you can grow some of those people internally and get them to that level, but at the very beginning, it's really valuable to have people who can play that role and bridge that chasm. So that to me is one of the really important piece of advice, not I mean, generically about building companies, but specifically companies of this type.
No, it's really phenomenal advice across the board, and I know we and our listeners thank you both. So shifting gears slightly and diving a little bit more deeply into AI for drug discovery, development, and the advancement of precision therapeutics, let's take all of this a step further and dive more deeply into that intersection of tech and bio. And as we think about applications of AI and ML and data to therapeutics, I think they can probably be best described as a spectrum, ranging from things like novel insight generation for drug repurposing, all the way to the holy grail of really truly in silico design and development from beginning to end. And I think I think we'd hopefully all agree that we are a ways away from that point, but hopefully we're also making strides. And so, Daphne, you recently spoke about recalibrating industry expectations of AI in drug discovery. Can you expand upon maybe some of those expectations here? What should we be thinking about in terms of applications of AI and ML to biology? Where are we today? How should we be thinking about timelines for what's coming next?
So I love the way that you frame that, and I'm going to start with presenting the two extremes of the perspectives, neither of which I think are the right thing for us to be thinking about today. The first one is the one that you alluded to, which is purely fully in silico. You start from, here's the disease that I'm looking to address, and here's a drug that comes out the other end. And I've heard some people talking about end-to-end machine learning for drug discovery, and I have no idea what that even means. I don't think we're anywhere even in the same solar system as that level of capability today. And I think when you use that language, it is destructive, because it sets up an expectation that as soon as anyone looks even slightly underneath the hood, is like, wait a minute, but that's not even close to what's going on. And it creates this mismatch of expectations and an inevitable disappointment that and I hate to say this, you can see the graying hair. I've been doing AI and machine learning for quite a long time. I've lived through several AI winters, and I can tell you that those AI winters are a direct consequence of hyperbolic promises that had no grounding in reality, that created that inevitable disappointment, a complete decrease in funding. When I went to grad school, you couldn't say you were doing AI. You had to say you were doing cognitive computing, because AI was a dirty word. And that was because it was on the tail end of one of those AI winters. So I think it's really important for us to be balanced and calibrated in the promises that we make. On the other extreme of the distribution are those people who are like, yeah, this whole AI ML thing is going to be like a, little widget, and it might improve, in the same way that will let whatever computer aided design or X-ray crystallography improve the certain tiny little segment of important perhaps, but of the drug discovery process. And my analogy here is that AI and machine learning are not like X-ray crystallography or computer aided design. It is like computers, as in it's going to improve every aspect by a wide margin. Is it going to be end-to-end? The computer magically intuits a new drug for ALS? Not anytime soon, but it is going to be transformative across every single step of the value chain. And some are further ahead than others. A lot of companies out there have focused, I think for obvious reasons, on machine learning for chemistry, because it's a well-defined input-output mapping. Input is a target, output is a molecule, and it's very easy to define whether you've met the criteria of the design constraints and such. I think what Chris and I are trying to do is a lot harder, because we're trying to actually uncover biology, which is a lot harder it's a lot squishier. It's a lot harder to, even figure out if you've actually solved the right problem or not. But at the same time, it's also where I think truly transformative breakthroughs are going to come, because most of the diseases that we're looking to tackle, we have no idea what the right target is. So it's not just a matter of identifying a better molecule. It's really identifying the right way to tackle the disease. And so I think that's an area of tremendous opportunity. But like I said, there is opportunities for this technology at every single step of the value chain.
Chris, you talked a little bit about how recursion, to Daphne, borrow your word, came from some of the squishiness of biology. Would love to hear your thoughts on the same question, and as maybe an expansion of what Daphne's been talking about.
Yeah, I think Daphne is exactly right. The truth is somewhere in the middle, and it's moving in a promising direction. When folks started working in this space, and I don't mean just biology or MLAI, but really the combination of these two together, it was universal, almost universal sort of condemnation of the potential of the space. And I think as we and others have made progress, there's now a greater number of folks who see the potential that I think Daphne and I know exists in the space, and many others do as well. But there's still a lot of folks who, even with some of the great breakthroughs like AlphaFold and others, still see this as very incremental. I think one of the ways I think about this is there's hundreds of steps to discovering and developing a medicine. And I've seen some really elegant point solutions to one or more of those steps. But most of the companies or even academic labs working on this, they tend to have focused, as Daphne mentioned, on the chemistry problem, for example, or there's companies focusing on predictive ADMET, or there's companies focusing on animal model translation or organoid type problems, et cetera. And the reality is we're going to need many of the solutions across many of these steps to create the kind of step function improvement that we all hope will happen over the coming decades. And so it's really about sort of compounding improvements across many, many steps. Now, why did we start with the squishiness of biology? I think Daphne nailed it there, which is the majority of drugs fail, and the majority of those that do fail because we pick the wrong target. Either the target doesn't do the thing we hope it does, or the target ends up doing something bad that we didn't realize it would do. And so the 0 to 1 shift in our industry, I think, is going to come from picking the right target or the best target at the beginning of a process before you've invested a lot of time and money and effort, and potentially even done experiments in patients, perhaps that are misdirected. But that will be insufficient. You're going to need improvements across so many other steps to ultimately achieve. I don't think we'll ever achieve in my lifetime at least, a perfect input-output of like enter disease for you and a pill pops out the other side 10 seconds later. It's not going to happen. But I do think over the 30 or 40 years in the future, it's going to be pretty incredible to see how far this field can shift. And I think we will see dramatic improvements over that kind of timeline in the probability of success of programs that are driven forward because of this compounding effect of all of these great solutions. And so the calling, I think, now for companies in this space, for people working in this space, is how do you start to think about integrating the best solutions? It's going to be insufficient to be able to just do good point solution A, B, or C. Ultimately, you're going to need to start combining these things. And I think if you look at companies, Daphne talked about doing work across not only sort of cellular biology, but also starting to bring in patient data. We're doing data aggregation at Recursion in our own wet labs at the cellular level, at the iPSC level, at the animal level with computer vision and machine learning on videos, and ultimately have ambitions as well to do it broadly across the different spectrum. And those are really going to be the kind of steps that I think are necessary to really start to eke out significant gains from the kind of work that we all are doing.
So as we think about that, as we think about building and combining these different steps and these different technologies, I think we'd all agree that computation, and here I'm using that as a shorthand for AI, ML, big data, data science, all the above, their applications to biology have a lot of promise, but to go back to what you were saying, Chris, about bringing programs forward, translation can at times be a challenge. And I mean that at every stage, as we all know, in vitro to in vivo to clinic. And so to both of you, how do you think about beginning to bridge this translational gap and really move and combine those steps to bring them forward? Daphne, would you like to start?
Sure. So for us, I think that this was a core problem that was in our mind as we set up the company, that that translatability chasm, if you will, between what you can do in wet lab experiments and what happens when you actually put the drug in a human. So for us, we're actually trying to go from that side all the way to that side. And the challenge, of course, with experiments in a human is that you don't get to do that until the very end, which is called a clinical trial, and it's very limited and very expensive, and by that point is way too late to affect your initial R&D efforts. And so the approach that we've taken, and it aligns with, I think, some of the growing success, if you will, in pharmaceutical R&D, is to rely on experiments of nature as a guidepost. And we've all heard the statistics that drugs that have a foundation in human genetics are twice as likely to succeed as ones that do not. And that is because there is effectively an experiment of nature that says, I twiddle this gene across the population, and these people ended up being healthier than those people, lower risk of disease, slower progression, whatever. And so for us, the idea would be that that effectively provides a ground truth on translatability. And of course, there's caveats to that, and I can speak to that. But to us, that's like an experiment that allows sort of preemptively do a clinical trial. Now, the challenge, of course, is that when you have a human population, you are at the mercy of the experiments that nature elected to put in front of you, which is the extent of the genetic variation that exists in that actual population. And that might not be the thing that you are hoping to do in terms of your drug, even if your drug is genetically based so that you're going after a particular genetic target. It may be that you don't have enough data to say what that genetic perturbation does in a human population. And so that's where what we do on the other side is that CRISPR-based lab that we described earlier, where you get to do the experiments in cellular systems, say, if I make this intervention, this is what happens at the cellular level. But that, of course, creates a gap, because on the one side, you have the ability to intervene and do the right experiments, if you will, but a readout in the wrong system. And the other one, you have maybe not the right experiment, but in the right system. And what we're doing is we're using human genetics and machine learning to cross that chasm and really bridge what we see in the cellular system with what we see in the human clinical outcome. And so that's really the premise for what the company does. And in the human genetics is really, in some ways, and I mentioned that in an earlier comment, is for us a North Star.
I agree with Daphne on all of those points. And I think you'll see a lot of companies in the space who have started, as we have, in the context of genetically defined disease across therapeutic areas, from rare diseases to oncology. We're focused on places where these experiments of nature have taught us something. And like Daphne, we're doing large-scale experimentation. We've knocked out every gene in the genome in multiple human cell types with multiple different CRISPR guides. And that sort of data is really combining the power of forward and reverse genetics. And when you put those two things together, you sort of get this multiplication of your ability to understand and explore biology. And I think what's key to that, and I think Daphne mentioned this, but just to make it extraordinarily explicit, is the creation of these iterative or virtuous cycles. This is a very complex set of problems we're trying to understand. Biology is squishy, as Daphne said earlier. And so being able to sort of create an unbiased hypothesis, test that hypothesis, learn, and iterate is really, really important. I do think this is going to be a challenge for companies who don't have either a wet lab, or if they're in the chemistry space, some sort of simulation capability. The static public data sets, I think, will be insufficient to answer many or most of the key questions. They'll be important pieces of the puzzle, but you really need to create the capability to iterate because it's very, very complex, these sort of search trees that we're going down. The other piece that I'd add, and I think is essential, is if you look back over the last few decades, there's been, I think, a realization that perhaps many of the models and systems that we use are overly reductionist. And I'm not talking about cells in a dish. They are, everybody acknowledges that, but I'm actually talking about the readouts. Often we do these unidimensional sort of phosphorylation site X goes up or down, and just random movements can confirm your prior bias half the time. And I think as we start moving into these omics data sets, where companies, where teams, academics are collecting omics data at every level, we're creating for ourselves much more depth and breadth and the ability to understand biology in a much more interwoven way, which is almost always going to be closer to the truth, right? This kind of like Nature Science Cell paper of like right when I started my PhD in the mid 2000s, like the prototype of that was like protein A goes to protein B goes to protein C, and maybe there's like a feedback loop. I think probably a lot of that is right, but it's missing the hundreds of other inputs and outputs that probably exist. And the tools that we're building, others are building, are giving us the opportunity to start to really sort of disentangle the complexity of those systems at a cellular level. Now, that's insufficient, but you can start to, again, layer these solutions on. And so I'll go back to our technology and animal models. I mean, this is a space where it's kind of incredible how little changed in that field for so many years. In the space of neuroscience, for example, even today, folks often will put mice onto a rod that slowly rotates and time how long it takes them to fall off, the rotorod experiment, which is a way to sort of test for cerebellar and other sort of neurologic function or gait. This is a very, very messy, noisy test. And when you start applying things like computer vision and machine learning to just monitor a mouse in its native environment, in its habitat, in a cage, what you actually see is you can pick up much more sensitive and specific signatures of disease, but also potentially of toxicity. So we started deploying this technology to look at our mice when we were first testing sort of the PK of our molecules. And typically people look at two things, like death of the mouse and massive weight loss as the red flag that would tell you, like, let's slow this program down and try and understand what's happening here. And when that happens, it's good that people slow it down and try and understand what's happening. But what we noticed was that if you applied these tools to monitor mice, you could see gross changes for certain molecules in behaviors, sort of their circadian rhythms, their temperature, their behavior around the cage, their social interactions, their breathing rates. All of these things would be changing in very significant ways that we didn't have a way to describe or understand with the mechanism of the molecules we're testing. And we've been able to actually flag molecules probably 3, 6, 9 months earlier for potential worry and dive deep on that problem, half a year or a year before we otherwise would have, because we're employing this more systems biology approach to understanding something even like the initial sort of PK experimentation in our animal models. And I think that's the beauty of these approaches when they start to harness biology's depth and complexity through sort of high dimensional data sets.
So that's a great segue, Chris, and it leads us to talk about our next focus on next generation data sets, enabling new modalities using some of these technologies. So as George Box once famously said, all models are wrong, as you were talking about, some can be reductionist, but some are useful. And so let's talk about the data sets that enable these discovery models we've been discussing. And Daphne, in the past, you've expressed great appreciation for biobanks, for example. And with the rise of biobanks that are generating really well-characterized genotypic, phenotypic, and EMR data sets, and Chris, going to what you were saying, needing to link across different data types, we're accessing more diverse patient populations, and we think that the heyday of biobanks is really only just beginning. So we would love your thoughts, Daphne. How should we think about biobanks today and how they could potentially be improved?
So first of all, I love that George Box quote. It's one of my favorite quotes. I think we've used it in half our board decks and all hands present, not quite half our all hands presentations. It's really I think an important thing for us to keep in mind. But coming back to the question, biobanks, I think, are incredibly important because they do create a broad-based and I would say in some ways hypothesis-free version of picture of human biology and human disease that allow you to come in and interrogate it in different ways. In this way, it's different from a clinical trial, that it's not just interrogating a particular patient population with a particular disease and a limited set of biomarkers that are specifically designed to capture the biology of that disease. It could be a very broad-based picture of human biology, both on the population level and on what one measures. And one of my favorite biobanks in this regard is the UK Biobank, because they actually successfully did both of those things. Now, admittedly, the population diversity is not as large as we would like because it was Eurocentric. They're trying to expand beyond that, but it was otherwise a fairly broad spectrum of individuals and an incredibly deep phenotyping for those individuals, ranging from all sorts of lifestyle covariates, life histories, and importantly, unbiased, high content measurements of those of the human biology, ranging from imaging of brain and abdomen and entire skeleton, blood biomarkers, urine biomarkers, and they keep expanding that. Recently, they did blood proteomics, which was, I think, very empowering and uncovering a yet additional layer of biologies. So if you look every week, there is 2, 3, 5 papers that uncover a fundamental aspect of human biology by looking at those high content data, which, by the way, are aligned with human longitudinal clinical outcomes via the tie-in between the UK Biobank and the National Health Service, which allows you to kind of see, as these people continue to age, how their cross-sectional health state, as measured at the intake time, ended up manifesting into ultimately what diseases they ended up having via their ICD codes. Now, let me highlight several aspects of what I think makes this biobank so special. So one of those is as I said, the very dense and unbiased phenotyping. There's a lot of biobanks out there that all they have is this, intake form and maybe some basic demographics, maybe some electronic health record stuff, but they never assay in a comprehensive manner the human biology. So that's one aspect that I think is really unique and I wish more biobanks would do. The other one, which I think is equally important, is the longitudinal follow-up, because there's only so much information one can get about disease if you look at a single time point. What you really want to understand is each person being their own control, how they progress into disease over time. And I think that is the other thing that this biobank did particularly well. The third thing, and I will pause there so that Chris can also speak, is the fact that this database is open to any qualified researcher with appropriate protections. And there's so many data sets out there as of today, including all of us, which is the US version of this, that are only available to a tiny, tiny fragment of researchers who can bring value from this. And to my mind, the thing that made the UK Biobank so special is that anyone is part of the democratization process that we were talking about earlier. It's allowing people with creativity from all over the world, as long as they're protective of privacy and so on, to really extract value for human health from this incredible resource.
I think Daphne really covered that nicely. Not a lot to add. Maybe the only kind of push or pull that I'd give there is, I think a component of this democratization has to be in bringing together and bringing along patients as a part of this journey. And I think as we start to demonstrate progress in the field, and as the world moves in a direction of sort of being more literate in the space of data and its value, I think it'll be very interesting to watch how the value of patient data is ascribed and to whom it's ascribed, and also to find those companies who are able to sort of excite people about their own data, and those companies that are able to involve people in sort of this giant step for humanity of us to understand biology more deeply. And I think UK Biobank is doing that in many ways, and there's other fantastic groups that I think are doing that as well. And ultimately, we're creating a sort of a whole generation or generations coming together that are going to push this field forward. It's not just going to be companies, it's not just going to be academics, it's going to require all these communities together.
I really like that last point, Chris, about all of these communities together. And I think that's one area, biobanks, greater representation of historically underrepresented groups will also go a long way. Thank you both. I'll pass it off now to Chaz to guide us through our third topic, the evolution of business models in tech bio.
Thanks, Chris. And we'd be remiss, folks, as we talk about tech bio, to not mention platform companies and the evolution of business models that have come with the space. Chris, we'd love to start with you here. Is there a mix of real business models here between partnerships, out-licensing, in-licensing, spin-outs, the full gamut? How should companies navigate this, and how should they think about kind of mix and matching different styles? I know that playbook's still being informed, and Recursion helped write a lot of the early days of it. We'd be curious to hear your thoughts.
Well, I think we've done everything on that list at Recursion at one point or another. The only thing we haven't done at this point is fee-for-service work, and we've learned a lot. And I would say that some of the things we tried in the early days, or the things that others have tried in the early days of this space, it taught us a lot, and also may not be the right things for companies to do today. And a lot depends on the technology that you have. In our context, back in 2013 when we founded Recursion, with a sort of core focus on biology first. Chemistry is important, but for us it was the biology first. We wanted to identify new opportunities for targets within the context of specific diseases, and in particular rare genetic diseases. And so repurposing, in-licensing these molecules was a great opportunity for us. But as we continued building, and we will continue to opportunistically in-license whenever we see a good opportunity, the reality is that the vast majority of potential targets in biology have not really been drugged yet, and that some of the most exciting targets for most of the diseases we're working on aren't ones that others have already made a drug for. And also that there's a ton of perverse incentives, both, I would say, talked about and not talked about in our industry, that minimize the probability of success of molecules that might be in-licensed. And so ultimately, we ended up building our own pipeline, both of repurposed drugs, and yesterday, or I'm not sure when this will air, but in early September, we announced our first new chemical entity entered a phase 1 clinical trial in Clostridium difficile colitis. So we're sort of bringing together both of those spaces. And we've also partnered. We're partnered with Roche Genentech in neuroscience and one oncology indication, and partnered with Bayer in fibrosis. And we think the beauty of a platform is that you can build a portfolio, not only of medicines, but a portfolio of business approaches, of economics, both programs that might bring in economics early for a company, others that might sustain a company later with higher royalties. And you can start to actually really create this sort of portfolio for your company beyond just sort of the medical side of it. And that's very critical. It creates a lot of opportunities as you engage with investors and others to lay out not only the short-term, but the long-term vision. And I think increase flexibility for companies like us. Daphne?
So I think you summarized it really well, Chris, and I'm not going to repeat all of what you said. I think there's just an incredible opportunity of diverse business models at different stages, and we've also benefited from some outstanding partnerships. I'm going to highlight one other aspect of partnerships that I think didn't come out in your comments, which is the access to data as part of partnerships. And we've had that benefit in 2 of our partnerships. One is in the early agreement we had with Gilead that gave us access to this incredible clinical trial data set where we learned and were able to demonstrate what machine learning can do in histopathology data and the kind of genetic insights that you get from a machine learning interpreted set of features on high content phenotypes. And I think that was really illuminating for us as well as for others. And then that really was in some ways the catalyst for the partnership that we recently had with Genomics England, which gives us access to this incredible data set that has histopathology, genetics, and coming up also blood proteomics that allows us to illuminate both oncology across the different types of oncology indications, as well as a diverse set of rare diseases. So I think there's a lot of business models and a lot of forms of value beyond just dollars that also come as part of those partnerships as well. And I think that's one of the really exciting things about being a platform is that you have all those different ways of partnering and all those different ways of value creation.
Thanks, Daphne. And Chris, one quick question for you. As we think about some of the founders in our audience today, in addition to navigating business models, a lot obviously to think about in the investor landscape, more traditional tech investors versus bio investors, East Coast, West Coast paradigms, and obviously thinking about exiting in public markets here. How should our founders think about navigating the investor ecosystem?
I could get myself in a lot of trouble here, so I'm going to be relatively nondescript in this answer. And I'll say what I tell a lot of founders in this space, which is not so much in the public markets, but especially in the early days of your private fundraisers. You are picking partners. And so don't worry about which coast they're on. Don't worry about whether they're tech or bio so much. Don't worry about how much dilution you're going to take in the round, nearly as much as you should think about, do you want to work with this person side by side every day? And some of our early investors, folks like Zavendar and Nan Lee, who were sort of the 2 major investors alongside DCVC in our Series A, ultimately were folks that I've spent more time with than some of the early employees at Recursion. And so that's the advice that I think is typically missing. There's tons of great advice out there about everything else. It's not that it's unimportant. It's just that you've got to recognize these are partners you're going to work with on hard problems. You're going to have to debate and disagree and still work together. And so you have to think of that. I think it's often deeply sort of underregarded in terms of its importance.
Absolutely.
And if I can just jump on top of that, and first of all, agree completely, and also say that especially in the biotech space, these folks are going to be with you around your table, not only before you go on the public markets, but also after you go on the public markets. And there's people out there who will happily dump your stock as soon as it's liquid. And then there's folks who will be with you for the long haul, because they don't care about making a quick buck. What they care about is really helping build transformative companies that create value in the long term. And we were able, both in our early, more venture-based rounds, but also importantly in our Series C, where we started to bring in more crossovers, to get the right group of people who are in this because they think it's a long-term transformative company that will create massive value. And it's not about how quickly can you sell the stock so you can bring back money to your limited partners. And I think that's a really important additional layer on top of, you know, be sure that when you're bringing investors around your table, it's not just about, you know, the valuation that you get or the term sheet and so on. It's about who do you want to spend the next, you know, 10 years with, and how will they treat you once that signature is drawn on the page.
Thank you both once again for joining us. One final question to each of you. Any closing thoughts, and how can our audience learn more about your work? Daphne.
Go ahead, Daphne.
I think this is an incredible time to be in this space. People often ask me, you know, you're a machine learning person. Machine learning can be used for so many different things and so many different areas. Why did you pick this one? And the answer that I like to, that really drove me is that I think that science comes in waves of innovation, and in certain periods of history, there's certain fields that just like are on a rocket ship trajectory and make a tremendous amount of progress in a short amount of time. You know, you think back on the late 1800s, that was chemistry because of the periodic table. Early 1900s is physics, 1950s computing, and the 1990s was actually kind of a bifurcation because data science started to emerge as a discipline in its own right, which is kind of related to computing, but not only, because it's also statistics and optimization and neuroscience. And then what I call quantitative biology started to emerge as a field where we suddenly started to measure biology at reasonable and increasing scale and fidelity and accuracy and so on. And those two fields kind of evolve in parallel. And what we're seeing now is that convergence. And I think this is a new field. And it's a field of data and biology combined, and I think it's going to be this incredible opportunity to make a big difference in the world. It's going to make a difference in the healthcare space, which is where we are, and both Chris and I, but also in the environment and energy and agriculture and so on. And this digital biology, which is what I call this field, I think is just an incredibly powerful place to be, to both do incredible science, but also make a very big transformative difference in the world. And you can find out more about what we do at insitro.com. Sorry.
I couldn't have said it better. I think Daphne really hit on all the high points. The only thing that I'd add is for those who are founding or building companies in this space, it's hard. Building a company and pushing a new kind of merging of industries forward is hard. And the best advice that I've ever gotten, and I hope the best advice that I can give, is when it's hard, go find a patient. Go find a patient to interact with. And if you interact with a patient, you recognize that when you're sick, all that matters is being healthy. And that's true for the patient, their family, and their friends. I think it can reinstill all of the passion and all of the energy and motivation. And so just keep yourself close to the patient, remember the patient, and as you're developing these medicines, make decisions that you know patients would be proud of as you take these medicines forward through discovery and development. And if you do that, whether you're successful or whether you fail, ultimately, I think we'll all be in a better place if everybody makes decisions in that way. You can learn more about our work at recursion.com, or you can follow me on Twitter, I guess.
Fantastic.
I don't tweet, so you can't really follow me on Twitter. Not really.
Thank you both again for your time, Daphne and Chris.
Thank you.
Wonderful episode.
Thank you.
Grateful for you joining us. Hope to have you back on the show. Thanks again.
Thank you.
Thank
Bye.
you. Appreciate it. Bye-bye.
Bye.
Thanks, folks. Take care.